Hello again. This time we're going to be talking about how to take DNA and make photocopies of DNA so that way each new cell can have some. When you have a cell, when you start out as a newly fertilized egg, you have one cell and one nucleus. And that cell needs to split and make two cells, each with identical sets of DNA, the same number, 46 and 46 to each offspring. And they need to split and so on until you are made up of trillions of cells. And each of those trillions of cells need to have the same exact DNA as the very first cell. So the only way we can do that is if we can do making copies of DNA accurately and without mistakes, because when we get mistakes we get mutations which can cause diseases and problems. So we're going to learn how that I can take my 46 strands of DNA, turn that into 92, so that way when I split my cell they each go back to 46. Okay, so we call that process replication, DNA replication. That is the process of replicating a strand of DNA into two identical strands. So here is my DNA strand. We refer to that as the parent strand. And then the new strands down here we call the daughter strands. I don't know why they don't call them the son strands. I have no idea. They call them the daughter strands. And so what we're going to do is we're going to take a look and see how I can use one half of my DNA strand to act as a blueprint or a template for the other half. And then what we'll get is two brand new strands of DNA. Okay, so the first thing that needs to happen in order for this to work is this enzyme called DNA helicase needs to come in. I know it's an enzyme because it ends in ASE, and the helic part refers to the helix. The helix is a fancy name that we give DNA, the twisty nature to it. So DNA helicase breaks the twisty nature of the DNA. It breaks it in half. So what he does, he kind of acts like a little plow, and here's my DNA strand. Here's deoxyribose, phosphate, deoxyribose, phosphate. Here's my different bases. You can see the hydrogen bonds, 2 and 3 bonds. And so what happens is DNA helicase comes in and breaks these bonds. When he breaks these bonds, that opens up the base. So that way now he's not attached to anybody. So you can see this guanine used to be attached to this cytosine. This cytosine used to be attached to this guanine. And so all I'm doing is breaking up the bonds between the two bases, allowing the bases to be free and open on the inside. This area where the DNA kind of looks like a Y or a fork in the road, we refer to that as the replication fork. So right here where my DNA is splitting, we call that the replication fork. Okay? And so we've got, here's our DNA strand. We see the sugars, the phosphates, and the bases inside being broken by DNA helicase is breaking the hydrogen bonds that hold them together. Next thing that's going to happen is another enzyme called DNA polymerase. Again, it's an enzyme because of the ACE, and the polymer means it's an enzyme that's going to build the DNA polymer. So what he's going to do, you can see him right here, and right here, and right here. He, on each side that's being ripped open, he is going to get on there and he's going to read which base is on the parent strand. So like right here we have this gray one. So we look down here and we can see that that kind of looks like it's gray and pointy, kind of looks like a guanine here. So he says, okay, that's guanine. Who goes with guanine? Cytosine. So he's going to find a cytosine and he's going to glue it onto this strand right here and let it grow. And then he's going to hop down to the next one. See, it's adenine. Thymine goes with adenine, so he's going to glue a thymine on here. And so as he does this, he's just going to work his way through and build a new half of the DNA. So on this guy, on this side, he just goes in order, 1, 2, 3, because DNA polymerase works in a particular order. If we call the upside, you remember how we said that they're upside down like this? We're going to call the upside 5'. Don't worry about what that means. We're just going to call up 5. and we're going to call down 3. And so that means over here, 5 is over here, and 3 is over here. Now, DNA polymerase only works, if you follow it down, it only works in a 5 to 3 direction. So on this one, he can go from 5 to 3 in order. But on this one, he has to go this way, 5 to 3. And so what happens is he goes a little bit until he bumps into the next one, and then he hops off and goes backwards and builds until he joins up, and then he hops backwards, and he keeps doing this like leapfrog kind of thing. So one side of DNA is being made continuously, the other side gets made in very small chunks that get sewn together. So DNA polymerase finds a complementary base and attaches it to the base on the original strand of DNA. So again, you can see here's adenine, here's thymine, adenine, thymine, guanine, cytosine, cytosine, guanine. So all he's doing is gluing together and making a new strand of DNA that is complementary or opposite to the original strand. And what that does is it makes what used to be there when the two sides were together. Because when the two strands were together, it looked just like this. So all he's doing is making two strands that look identical to the original. Okay, next thing that's going to happen is that DNA polymerase is going to continue doing its thing, unzipping the DNA, and then polymerase is going to come right along and build the new strands until they run out of room. Now, since DNA is very long, what ends up happening, instead of DNA polymerase working on one end and going all the way down to the bottom, it usually makes these little bubbles. And so that way it can do it all down the length of DNA at the same time. So if this is my DNA strand, we'll see that we've got little polymerases all over the place, and they're doing their job, making little bubbles in here. And so that way it can go by much quicker, because if I have one going top to bottom, it's going to take a long time. But if I have a whole bunch of them, each doing a small portion, it'll happen much quicker. So what we see is that instead of going in one direction, we have the little replication fork going in two directions. And this bubble is just going to get bigger and bigger and bigger until they finally reach the ends. And so you can see here that my DNA is being made in this direction and in this direction. And these two sides are going to be absolutely complementary to each other. Now, this type of replication that we see here, using one half to make the other half, we call that semi-conservative replication. Semi meaning half, conserve means to save. So what's happening here is that we are saving half of the original DNA every time we make a new one. So here is your very first strand of DNA. It rips in half. Here's half one, here's half two. And then we can see the new half is being made from it. So red is new. So I have one old, orange, one new, red. So half is saved, semi-conservative. And then when he splits, the orange one's going to go here, the red one's going to go here, and now green is new. So orange is old, green is new, red is old, green is new. And then they're going to split. The orange and the blue are going to become the olds, and the black will be the new. The red and the turquoise will be the old, the black will be the new. And this just continues on and on and on until you die, basically, and then your cells don't need to replicate anymore. So we call this semi-conservative replication because half of the DNA strand is saved in every new copy. That would be like, instead of photocopying on a photocopy machine, where I take my original and I put it on a machine, the photocopy machine, and I have my old paper and my new paper. It's not like that. What happens in semi-conservative is that I take my paper and I cut it in half. So now here's what happens. I take half and half. So I rip my paper in half like this. And instead of photocopying the whole thing, I only photocopy the other half, like this. And I still have two new copies. They're identical to each other, but you can see I have saved half of my original piece of paper. So does that make sense? Different than photocopying, where your old one stays intact. In here, half is used as a template for the other half. Okay, that's it for today, and I'll see you next time. Bye.